Good day, everyone. Welcome to my channel. In today's session, we will be discussing an essential topic in human biology, the role of the blood. Blood is a vital fluid that sustains life by transporting gases, nutrients, and waste products, while also playing a key role in defense and regulation. Let us begin by exploring its remarkable functions. The cardiovascular system is the mass transport system in mammals. It is composed of three main components: the heart, which functions as the pump, the blood, serving as the transport medium, and the network of blood vessels through which circulation occurs. Blood circulation delivers essential materials to body cells and removes metabolic wastes. The exchange of substances between blood and cells occurs mainly through diffusion and active transport. Capillary walls are just one cell thick, allowing a short diffusion distance, and every body cell is near a capillary. Blood, the transport medium of the cardiovascular system, is over 50% plasma, its fluid component. Blood plasma is a pale yellow fluid. Plasma carries digested nutrients from the intestines or storage to cells, transports waste like carbon dioxide and urea to lungs and kidneys, delivers hormones to target organs, distributes heat to regulate body temperature, and helps stabilize blood pH. Red blood cells number about 4 to 6 million per cubic millimeter of blood, 4–5 million in females, 5 to 6 million in males. Produced in the bone marrow, they contain hemoglobin, the red pigment that carries oxygen. Mature RBCs lack a nucleus, providing more space for hemoglobin, and have a lifespan of roughly 120 days. Red blood cells transport oxygen from the lungs to body cells. Their biconcave shape provides a large surface area for rapid diffusion. Each cell contains 250 to 300 million hemoglobin molecules, carrying about 1 billion oxygen molecules, and also helps transport some carbon dioxide back to the lungs. White blood cells are larger than red blood cells but flexible, allowing them to pass through tiny vessels. Numbering 4,000 to 11,000 per cubic millimeter of blood, they are produced in the bone marrow, with some maturing in the thymus. They contain a nucleus and colorless cytoplasm, with some showing stainable granules. White blood cells defend the body against infection and play a key role in the inflammatory response to tissue damage. Hemoglobin is a large, globular protein composed of 4 peptide chains. Each chain contains an iron group, allowing a single hemoglobin molecule to carry up to 4 oxygen molecules. When bound to oxygen, it forms oxyhemoglobin, a reversible process that enables efficient oxygen transport throughout the body. The structure of hemoglobin enables cooperative binding. It contains 4 hem groups, each with an iron ion, Fe², capable of binding a single oxygen molecule, allowing efficient oxygen uptake and release. The first oxygen molecule binds slowly because the hem groups are hidden. This binding changes hemoglobin's shape, exposing the other hem groups and making it easier for more oxygen molecules to attach. In cooperative binding, the second and third oxygen molecules attach more easily than the first, while the fourth binds hundreds of times faster, demonstrating hemoglobin's efficiency in oxygen uptake. When oxygen binds to hemoglobin, it is quickly removed from the free oxygen in the red blood cell's cytoplasm, keeping its concentration low. This maintains a steep diffusion gradient from the lungs, allowing oxygen to continue diffusing into the red blood cells until hemoglobin is nearly fully saturated. Oxygen dissociation curve: at low partial pressures of oxygen, such as in respiring tissues, hemoglobin's affinity for oxygen is low. Even a small drop in PO triggers a large release of oxygen, ensuring that tissues receive the oxygen they need for respiration. Hemoglobin is nearly fully saturated. At medium oxygen levels, cooperative binding is strongest. When the first oxygen molecule binds, hemoglobin changes shape, increasing its affinity for more oxygen. As a result, a small rise in PO causes a large increase in hemoglobin saturation. At high oxygen levels, like in the lungs, hemoglobin has a very high affinity for oxygen and becomes nearly fully saturated. Even if PO rises further, saturation plateaus, forming the top of the oxygen dissociation curve. Biological importance: in the lungs, where PO is high, hemoglobin loads oxygen efficiently to reach maximum saturation. In respiring tissues, where PO is low, it releases oxygen effectively to supply cells. The steep middle of the curve allows hemoglobin to respond quickly to small changes in oxygen levels, ensuring active tissues receive more oxygen, especially during exercise. Carbon dioxide produced by respiring tissues diffuses into the blood down its concentration gradient. About 5% dissolves directly in plasma, 10 to 20% binds with hemoglobin to form carbomonohemoglobin, and the majority is carried as hydrogen carbonate ions in red blood cells. Carbon dioxide reacts with water to form carbonic acid, which then dissociates into hydrogen ions and hydrogen carbonate ions. This reaction is accelerated by the enzyme carbonic anhydrase. Carbonic anhydrase plays a key role in carbon dioxide transport. In tissues with high CO, it speeds up the forward reaction, producing more hydrogen carbonate ions. In the lungs, where carbon dioxide is low, it catalyzes the reverse reaction, releasing carbon dioxide to diffuse into the alveoli. Free hydrogen ions can make blood acidic, but hemoglobin acts as a buffer by binding to these ions to form hemoglobin-H acid. This helps maintain a stable blood pH and supports homeostasis. The chloride shift occurs when hydrogen carbonate ions move out of red blood cells into the plasma, and chloride ions move in to maintain electrical neutrality. The Bohr effect describes how carbon dioxide and hydrogen ions influence hemoglobin's affinity for oxygen. It explains how hemoglobin efficiently loads oxygen in the lungs and unloads it in tissues depending on the local environment. In active tissues, high levels of carbon dioxide produce hydrogen ions, lowering blood pH and slightly changing hemoglobin's shape. This reduces hemoglobin's affinity for oxygen, allowing more oxygen to be released where it is most needed. In the lungs, low carbon dioxide levels mean fewer hydrogen ions and a higher blood pH. Hemoglobin's shape favors oxygen binding, resulting in maximum oxygen uptake. Physically, the Bohr effect ensures oxygen is released in active tissues with high carbon dioxide and low pH, enhances oxygen uptake in the lungs where carbon dioxide is low, and aligns oxygen delivery with tissue demand. The fetus depends on the mother's oxygen supply through the placenta. Maternal blood flows close to fetal blood, and fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin. This allows fetal hemoglobin to take up oxygen from maternal blood even at relatively low oxygen levels. Fetal hemoglobin ensures efficient oxygen uptake from the mother's blood, and the countercurrent flow of maternal and fetal blood maintains a steep oxygen gradient for maximum transfer. Thanks so much for watching. Stay tuned for more videos, and don't forget to hit subscribe, like, and share. You can also check out more resources on my website at www.edexcelbiology.com.